My talk is going to be about, as Joey mentioned, switching patients from established replacement therapy to extended half-life therapies, and I'm also going to talk briefly about emerging treatments. So those are my disclosures. For years, we've had a whole range of factor concentrates available, standard half-life therapies, and they're all really remarkably similar. So it's a bit like going into a cake shop and choosing fairy cakes. They're all really similar, but they've got slightly different toppings. But basically, you know what you're getting. You're getting a fairy cake. The issue now with the new extended half-life treatments is that the cake shop has changed, and there is a whole range of new things that are out there. Boston brownies, chocolate brownies, some rather elegant-looking chocolate cakes at the front. And the question is, which one are we going to choose? Are we going to have a fairy cake, or are we going to have one of these new chocolate cakes? Are the new chocolate cakes potentially too filling, or are the fairy cakes something that we're used to? The issue here is that it's led to a certain degree of confusion amongst people in terms of what do you choose and how do you choose it. So, what I'm going to try and do is take us through some of the issues that might help us consider what we do and how we do it. Firstly, does technology highlight any issues that we need to think about? And when we think about the clinical trial data with the new therapies, does this show any differences that we need to be aware of? How comparable is the data from one trial to the next? Can we make those direct comparisons when we're choosing and considering which one or not? Health economics is a key issue, and obviously that's something that we need to think about. What are the issues there that are going to focus in? And of course, from a clinical and patient perspective, who do we want to switch? Should we move anybody? Should we move everybody? Or is it just some people that are going to benefit? And finally, how can we get the most out of moving our patients? And that's really something about pharmacokinetics. Now, obviously, the main issue when we're talking about improving clotting factor concentrates that we're talking about today is extending the half-life, so essentially prolonging the efficacy. So let's think about the technologies that do this. How have we been able to extend half-life? Well, as I'm sure you know, there are two basic mechanisms. And the first one is attaching a hydrophilic polymer conjugate to a protein to extend its half-life. And the one that has been used most frequently and is most furthest forward for hemophilia is obviously polyethylene glycol. So polyethylene glycol is a branch structure of various sizes which increases the capacity of clotting factors to remain in the circulation by increasing their effective molecular weight and above the renal threshold and becoming a big hydrophilic cloud. The second mechanism that we can use is by attaching another protein or structure which can then capitalize on other mechanisms that are around to extend half-life. And that is essentially the albumin fusion approach or the FC fusion approach, which both rely on the recycling mechanism of FcRn. So with both these constructs, they're taken up by FcRn, recycled through cells and put back into the circulation to extend half-life. So we have these two approaches, and we have different drugs, different hemophilia treatment modalities using both these approaches to extend half-life. There are, however, a couple of thorny issues that come up when we think about the technology, and unfortunately they mainly focus on the PEG technology. So I'm just going to address those and see whether they're a big issue that we need to think about or just something we need to be aware of. The first thing about PEG that everybody talks about and is concerned about is the metabolism and toxicity. PEG is not generally a very easily biodegradable entity. It's cleared by oxidation of alcohol groups, which is a fairly minimal process, or by urinary clearance, which is quite dependent on molecular size. Or if we use large PEG moieties, it will be dependent on biliary clearance or clearance by Kupffer cells. Because it's cleared by these mechanisms, we're worry about potential toxicity because it's non-biodegradable. So what is the toxicity data with PEG? Is there anything to tell us that it's not good? Well, I don't think there really is any strong evidence about it. Animal models have looked at the toxicity of PEG, and they found no evidence of toxicity after 2 years of extensive treatment. You only get acute toxicity with PEG if you give very large doses. And as you can see there, 10 grams per kilo is a huge dose. If we think about the therapeutic index, PEG has been shown to have a large therapeutic index, and it's around 600. So there shouldn't really be any metabolism or toxicity issues, but it is something that we do hear a lot about, and we need to be aware that this is a point for discussion. What about clinical data from PEG so far? There are many PEG-associated drugs that are out there, and there has been to date no evidence of PEG-related issues. But questions swirl, particularly in the pediatric field, about the issues of chronic use of PEGylated proteins and what actually happens. We can't eliminate it effectively, rapidly. It is a non-biodegradable polymer. And will that affect the whole body distribution or accumulation of PEG after repeated dosing for many, many years? The most we've used PEG in human studies, in human use, in therapeutics, is around 3.5 years at the moment. Potentially here we're talking about a lot longer. So the theoretical questions to raise. And we know now that there are also possibilities that it can be taken up and held in vacuoles in cells that have low or no turnover. So how does that affect us? Well, we can give an injection of a protein that contains PEG. As I've mentioned, some of it is cleared through the kidney relatively quickly for small molecules, more slowly for large molecules. For the larger PEG forms, they can be taken up in particular cells, and then they can form into PEG vacuoles, which are only slowly excreted and metabolized. What is the issue with this? Well, the truth is that we probably don't know at the moment, but it is something that people focus on and want to discuss. There is also the issue of anti-PEG antibodies that can be found in healthy subjects because PEG is ubiquitous, although we obviously consider it as non-immunogenic. Studies have been done to see how commonly these occur and whether they're associated with clinical hypersensitivity reactions. How common anti-PEG antibodies are varies depending on how you measure them, but there is probably no pathology associated with these antibodies. What's the relevance? The relevance is we need to be aware of it. One of the other issues I'd like to talk about when we talk about EHLs is what actually is an extended half-life treatment and what is a standard half-life treatment. I think most of us would accept a standard half-life treatment is about 12 hours. I think most of us would accept an extended half-life treatment, or a very obvious hair extension, at 20 hours. At 18 hours, I still think we can see that the hair is quite a bit longer, and that's extended. But what about 14 or 15? Is that an extended half-life treatment? The question has arisen, and there's been a lot of debate about what identifies extended half-life therapies. And there's now been papers recently produced to try and look at what consensus might be of what an extended half-life therapy is. And it's, by consensus, a bioengineered product. But we do need to think about products that may have extended half-lives but are not bioengineered. Are we going to draw an absolute black and white line there, or are we going to say what EHLs really refer to bioengineered products with a comparator arm that show they extend over standard half-life? So if we think about bioengineered extended half-life therapies, there are a number available to us now, which I've shown graphically on this slide. So we have a lot of these products, and they've been studied in clinical trial. How comparable is the data for all these products? I think one of the things that we need to look at first of all, and I'm not going to go through this slide in detail, but you might want to just scan through it, is you can see that for all the different extended half-life treatments here, the clinical trial protocol for the phase 3 is different. It is broadly similar, but it's not exactly the same. And so there is an issue about comparing one set of clinical data to another because the trial designs were different. But if we look at the clinical data, what can we see? We can see that in terms of relative increase, they all have a relative increase over standard half-life, although the mean half-life is somewhat variable. And at the end, I've put in a non-modified factor 8, which also quotes a longer half-life than standard half-life, but doesn't have a comparator arm. Why are these all relatively similar? Well, I'm sure we all appreciate now that this is because the major controlling element for half-life of factor 8 is actually von Willebrand factor, because 95 to 98% of it is circulating with factor 8, and that is controlling it. So what about clinical data? We know that they all extend to some extent. What about clinical data? There's a lot of data on this slide, and as I pointed out, as you'll see from the bottom line, the clinical protocol and changes to treatment in the trials were different. Some were fixed, some were investigator-determined, some were based on lab results, and some were based on clinical protocol. But if we look at the net effect, the ABRs, we can see that they're broadly similar in the range of 1 to 4, which is what we're becoming increasingly expectant for current treatments. So they all work in a prophylactic setting, there's no doubt. But can we strictly compare one set of results to the other, where the clinical trial design was somewhat different? I think that is more of a moot point. In terms of safety, all these products are safe, and we really haven't seen any significant signals in the phase 3 studies in patients that are more than 700 treated. What about factor 9? Well, we have 3 programs here, 3 factor 9 treatments that are available to us using different methodologies, 2 using FC recycling and 1 using hydrophilic conjugates in the form of glycopegylation. And here we see, unlike factor 8, a dramatic extension in half-life, up to from 82 to 102 hours, compared to standard half-life factor 8. What's interesting here, though, is when we look at the ratio EHL to SHL, you can see that it varies between the different therapies, and that's because the standard half-life factor 8, which in all cases benefits here, comes out at a different relative baseline value. And for 9FC, that was because the calculation for the half-life for Benefix was different to the calculation applied, or the pharmacokinetic model applied, to 9FP and 9GP. We can also see that the recovery is slightly different as well, with factor 9GP having a recovery of 2, implying that it remains essentially in the circulation. Whereas we know that 9 and 9FC, with a lower recovery, are potentially getting out of the circulation as well. So all these products exhibit significantly longer half-life, reduced clearance, and importantly, they are safe with no drug antibodies. Let's come on to the trial design here, because I think this is even more acute for factor 9 therapies than it is for factor 8 therapies. Here we have the 3 programs, and we have the 9FC program, which was based on maintaining a particular trough level and altering the dose or the frequency accordingly. 9FP studies, where the patient started on a fixed interval and then, depending on clinical response, moved on to a more extended approach. Or 9GP, where there were 2 simply fixed doses. When we look now at the clinical data, I think it means that it becomes quite difficult when the clinical protocol is so different to make big judgments about differences in ABR, which again are relatively similar, but it's hard to say that one is better than another because the situation in which they were trialled is significantly different in terms of clinical trial approach. But what we can equally see is that they are all effective and they all work. However, I would like to bring your attention to one particular point, which is the trough levels. For 9FC, this is 1 to 3. For 9FP, you can see that the trough levels were a mean of 20 or 12, depending on the dosing schedule. And for 9GP, 8.5 and 27. But what we might also see as an issue is that if we particularly look at 9GP here, we can see their trough levels, their mean trough levels, and we can see that they're substantial and look impressive. But we can also see that we do actually still get bleeding at these rates, and particularly at a rate of approximately 30 for a mean pre-treatment factor 9 level, one might expect an ABR of lower than 1, given what we know about gene therapy, and similarly with a trough level of around 12 at the lower dose. And that does imply that bioengineered products that may have different vascular distributions or may behave differently may be slightly different in terms of how we can interpret trough levels. So clinical efficacy and trough levels may be dependent on distribution. And I think it's important that we are aware that we can't necessarily take all that we know as received wisdom historically and apply it to bioengineered products, and we must look at it in the context of the data that the clinical trials show us. Moving on to health economics, clearly these products are all going to have to be marketed and priced, and it's important to consider what economics can tell us about how we're going to use these therapies. All the EHLs offer an opportunity to reduce the frequency infusion, but to maintain an equivalent trough. And as I've mentioned, it's difficult to compare product to product because there are no head-to-head trials. So how are we going to look at these in terms of a health economic approach? Is there a simple multiplier that we can apply, or is it more complicated? Clearly, there's a lot of work needs to be done in this area. We ourselves, though, did some work based on the hypothesis that treatment frequency would be the driver and looked at relative potential cost modeling using lower frequencies or the same troughs or the same accepted ABRs. And we based this on a hypothetical model with a patient with severe hemophilia A. Now, this slide shows an awful lot of figures. I don't want you to look at it in too much detail, but essentially we looked at different opportunities, different cases, different approaches of how you could use an extended half-life factor 8. And what we showed was that depending on the particular approach, we found that the extended half-life factor 8 could differ from the standard half-life factor 8, or be priced more if the figure is greater than 1, or be priced less if it's under 1, depending on the scenario that we used. In short, though these results are hypothetical, what they do reinforce is the idea that in most healthcare economies, that factor 8 EHL use, and indeed factor 9 for the same effect, is going to be highly influenced by cost. And it's the precise nature of the treatment regimes that they replace that is going to have most influence on how we use them. Taking this further, we can equally see how if there are significant price differences between standard half-life therapies and extended half-life therapies that exceed those price factors that we saw on the previous slide, those could have an undue and extremely strong influence on treatment options that might be available to our patients and in our own health economies. And indeed, we know that there is a lot of payer oversight in terms of using these new treatments, and this is a UK document looking at how we could employ EHLs, and as you can see, it's a very detailed document controlling, to some extent, our choices. At the moment, there are a limited number of EHLs, but as you would see from one of my previous slides, there are a lot more coming on stream. Will that develop more of a challenge for pricing, give us more opportunity and more patient choice and treater choice to allow us to swap patients over? Or is it simply just going to be cost that we end up thinking about? I hope that's not the case. What we have at the moment is historically lots of different factor concentrates that all did the job. We now have new factor concentrates, which are the next generation. They all do a better job, but they're a bit different. And which you choose is, to some extent, down to preference. One thing I'd just like to mention, though, before we move on to other topics, is that there is a future out there, and though we've got EHLs today, we will potentially be having newer generation treatments that will change the paradigm again in the concentrate setting. And I know that we're going to hear more about one of these products in the late-breaking session. So, when it comes to moving patients over, how do we decide what we're going to do, who we're going to move, and how do we make the choice? Well, some of us may feel that we have personal anxieties or worries about one treatment over the other. Some of us are going to have personal experience and familiarity and hands-on experience with one treatment over another, which is also going to influence things. But I do think that one of the issues that may play out for many of us in many of our treatment scenarios in the future is going to be cost, tenders, and competition. What about choosing, though, which patients are we going to put on? Who do we put on factor concentrates that are the next generation? How do we choose? Well, clearly, we can reduce the treatment burden by reducing the number of infusions. We can also use these treatments to raise the trough level to give us greater protection against bleeding. And I'm going to come back to another point that, paradoxically, these treatments may be particularly valuable for patients who are outliers. We also are going to find out from our multidisciplinary teams which patients might be most interested. We're going to have patients come to us in our clinics to ask about these treatments, and we're going to also find that they can be particularly valuable and useful in difficult or unusual settings. I think, in short, everybody potentially has something to gain from the extended half-life therapies, irrespective of the above, because in the vast majority of people, the treatments are going to last longer. Let's look at the outlier population. What we've talked about at the moment with most things is the population that sits in the middle. But these two outlier groups can get significant advantage with extended half-life therapies. If your half-life is very short and you have to be on daily treatment, if you can just extend it to bring it up into that normal range, you can get onto less frequent treatment and reduce your treatment burden. Equally, if you live at the high end of the range and your extended half-life therapy lasts even longer, you could get more clinical benefit by being able to both reduce treatment frequency and still get higher trough levels. It's not all about reduced frequency, and I think we need to recalibrate our thoughts and expectations in terms of reality and what we can actually achieve here, because it's more than just treatment frequency. So when it comes to switching, it's more than frequency, as I mentioned, and we need to think about all the things that we're trying to do. Is it frequency? That's number one on the list, but is it about getting a better trough? Is it about dosing and reducing less, or is it about all three? And how are we going to do that? How are we going to estimate the dose and frequency? Are we just going to do it by clinical monitoring for bleeds or following up trough levels? We've thought about this in the UK, about how we could employ extended half-life therapies logically and on an evidence base. And we drew up a set of parameters, and the first was a full pharmacokinetic analysis so that we got all the data that we needed. And we thought, that's probably not the easiest thing to do. Now, clearly, a lot of people have been thinking about this, and we now have new ISTH guidelines, which will allow us to get pharmacokinetic data without a washout, without fixed dosing, from relatively sparse sampling through a single dose, or even potentially through multiple doses. And this was certainly a lot easier to collect the data, and it means that we can then treat our patients on the basis of an evidence set of data. It's important to base it on real PK because just looking at 3 numbers can give you a different pattern of results. And I think that the latest article that's come out from the SSC, which is online first, just a couple of days ago, promotes the idea that pharmacokinetics, if we're swapping, is an ideal approach because it gives us the information that we need and allows us to maximize and optimize opportunities for our patients when switching. Sorry, I just skipped back. One of the programs that is highlighted in the SSC guidance is the WAP service, which is globally available and run out of McMaster and can give you data from a pharmacokinetic Bayesian analysis to support treatment, dosing, and individualizing for patients. I think it's moved beyond the opportunity now of just being able to have 2 out of 3. We can now, if we've got the evidence base and the ability to individualize, have all 3, not just 2 out of 3. And I think that allows both the patients to get the most out of treatment and us as treaters to get the most because we can achieve all the clinical aims we want and potentially reduce treatment burden. Now, as I mentioned, our cake shop is full of lots of goodies, lots of new things that are really exciting, but the issue is now that there is a new kid on the block, the macaron, and lots of other delightful cakes there. And this is our emerging therapy portfolio. Bispecific antibodies, RNA silencing, anti-TFPI, gene therapy, even potentially altering other clot suppressors. So there's a whole range of new treatments that are coming out there. How are we going to take this in context as well? This, I think, offers and throws up a whole set of new questions when we are going to have to think about switching. What sort of levels of unmet clinical need are they, and how does that marry up with convenience? Which is more important, convenience or unmet clinical need? We need to see really granular data on patient groups so that we can make intra-individual comparisons and see if we're ending up having to trade perhaps burden for bleed rate or not. We remain to see that, and clearly later today we're going to see some exciting data which will allow us to begin to answer some of these questions. We also need to bear in mind that there are going to be 2 perspectives here for these emerging treatments, a treater's perspective and a patient's perspective, and we must marry those up. And of course, something that we all know all the time, it's always about safety. We can't just go for convenience if we've got to bear in mind safety. Having said that, it is an exciting and hugely transformational opportunity for the future that we're looking at down the line. So in summary, I think if we just think about EHLs, that's a huge opportunity for incremental improvement. And in practical terms, all these products are effective. They do what they say on the tin. They're good for bleed control, and they're good for prophylaxis. I do think, though, personally, it's almost impossible to say that one product, one EHL, in any of these fields is better than any other one, because the trial design really profoundly limits product-to-product comparison. We are not doing the standard thing that we should do in all hemostasis, which is compare like with like. There are theoretical concerns voiced, but there's no real data to support that to date. We do need to bear in mind that the whole cost issue, marketplace and reimbursements, potentially in some areas can be huge impact in choice and the impact on what we have available to us to treat. And everybody has some benefit to gain. PK is going to be hugely helpful in achieving this. But the last thing to say is that having just gone through all of that and thought it through and got our evidence base and knowing how we're going to deal with it, we're now looking at a new generation of treatment which will throw this all up in the air again and make us have to think about changing things yet again. So, having talked about the cakes, having talked about the treatments, you probably would like me to tell you what my favourite is. It is the blackberry sundae and the millionaire cheesecake. Thank you.